Okay, we're going to go ahead and get started. Thank you so much for coming, everyone, in person, and those of you on Zoom. I'm a little scared by looking at the number of slides that are in this, but it'll be great. It's all good. So, hi, everyone. I'm Chris Gunter. I am an investigator in the social behavioral research branch, and I am extremely excited to be hosting my friend Sarah Tishkoff today. I first met Sarah at a poster session in 1993 at ASHG, actually. And had no idea that eventually someday in 2025, I would be on the board of directors and that she would be the president-elect. So blessed for trainees, talk to other people at poster sessions because you never know what will happen. So I've been a fan of Sarah's for a long time. So she is now, as she will tell you, a professor in the departments of genetics and biology at the University of Pennsylvania. She also is, I'm going to make sure I get the name right, the director of the Penn Center for Global Genomics and Health Equity in the Department of Genetics there. She's done amazing work, and she's going to tell you about a fraction of it today, but I encourage you to keep looking up more of her work. She's also been elected a member of, separately, the National Academies of Sciences and Medicine and AAAS, and has many, many awards to her name. But I am going to turn it over to Sarah and let her tell us about it. Thank you, Sarah.
Thank you so much for that nice introduction. I have to say, it has been so much fun to be here today and to talk with everybody, and particularly the lunch with all the students and postdocs. Awesome. So as Chris said, you're going to hear about a fraction of the work that we're doing, just talking about a couple of stories. But first, let me start. I guess I have to do it here with some of the key challenges in human genomics research that my lab is focusing on. So one is that we need to do a much better job characterizing both genomic and phenotypic variation in ethnically diverse populations. We want to understand the evolutionary processes that generate and maintain that variation, and to understand how gene protein, and gene environment interactions contribute both to normal variation and disease risk. The focus of my research has always been on Africa, and there are a number of reasons for that. One of which is the fact that all modern humans originated in Africa. These red dots represent the locations of fossils of anatomically modern humans, the oldest of which is dated to about 300,000 years ago. But we don't know really anything about when and where exactly modern humans arose in Africa, because there's a major bias in terms of where we find these fossils. So what we do know is that after this origin in Africa, say somewhere around 60 to 80,000 years ago, relatively small numbers of people migrated out of Africa, giving rise to populations across the globe. We now know that when modern humans left Africa, they ran into archaic populations like Neanderthals and Denisovans, with whom they interbred, such that somewhere between about 1% to 6% of the genomes of non-Africans derive from those archaic populations. So you can imagine this demographic history has really shaped the pattern of variation that we see in global population. The other thing that motivates our research is understanding risk factors for health disparities. So hypertension, for example, is more common in individuals of African ancestry. Adult onset type 2 diabetes, more common in people of Native American ancestry, also African ancestry. But as I think everybody's aware, there are major social, demographic, dietary factors that are playing a role in these health disparities. And I think but that does not rule out the possibility that there are also underlying genetic risk factors. And one of the biggest challenges today is distinguishing between the genetic and the environmental factors that play a role in risk for disease. So as we published in this commentary several years ago now, this was looking at the GWAS catalog, and this hasn't changed. If anything, it's actually gotten worse recently. They keep increasing numbers, but relatively more Europeans. So about 80% of individuals now, might be closer to 90%, included in GWAS or European ancestry. About 10% are of Asian ancestry, about 2% African ancestry, majority would be African Americans, 1% Hispanic or Latin American ancestry. Let's move that out of the way. And that's not good for a number of reasons. One is that it's going to impede our ability to fully understand genetic architecture, genetic risk factors for human disease, and it could actually make health inequalities worse. Our ability to translate genetic research into clinical practice or public health policy could be incomplete or even possibly mistaken. It could result in inaccurate assessment of pathogenic variants in clinical genomic studies. One of the reasons being that one of the criteria for determining if a variant of unknown significance is pathogenic is how common it is, with the idea being that if it's common, it's less likely to be pathogenic. Maybe it's not common in Europeans, but maybe it's common somewhere else. So that might actually change the classification. And as I'm sure you've heard, attempts to use estimates of genetic risks, so for example, polygenic risk scores, that are usually constructed from big, giant GWAS, predominantly in European populations, usually result in inaccurate assessment of risk when you apply these to individuals from diverse ancestries, and therefore lack of appropriate interventions. So as I said, my research focuses on Africa. Just to give you a little bit of background, there are over 2,000 languages spoken in Africa. These have been classified into 4 major language families. In blue, in North and Eastern Africa, you're seeing the distribution of populations that speak Afro-Asiatic languages. So that would include Cushitic and Semitic, for example. In red is the distribution of people who speak Nilo-Saharan languages. Many of them practice pastoralism and dairying. They're mainly in Central and Eastern Africa. In the sort of orange-yellow color, we see the most widespread language family, which is Niger-Congo, which originated in Western, Central, and Western Africa. And the largest subfamily of those languages are the Bantu languages, which are thought to have arisen around the region, sort of the border of Nigeria and Cameroon. Now, populations, Niger-Congo speaking populations, very early on, I think it was probably around 6,000 or so years ago, had developed iron tool technology and something called slash and burn agriculture. So they're able to cut down the trees in the forest. They're able to grow crops. If you have crops, you can sustain large population sizes. They were very successful and migrated across Africa to the east and to the south and to the west and to the south, where they then admixed with local populations and have really shaped the genomic landscape in Africa. And then in green, we have populations that speak with clicks that have been classified as Khoisan, the San in Southern Africa, and then 2 groups, the Hadza and the Sandawe, who live in Tanzania. So none of this work is possible without my collaborators in Africa. So I'm just showing you who they are. And so everything that I'm going to be presenting is done together with them. Just to give you a little bit of background of what we're doing, we're so far have been focused more on populations that are living in more rural areas, practicing more traditional lifestyles. It can be very challenging to get to these populations. So we're using 4-wheel drive vehicles, and we are having to bring all of our supplies with us. Trying to move that arrow also off. There we go. All right. Now, before we even begin this research, there's a number of things that we do. So one is you have to get IRB approval, let's say at UPenn. But what takes a very, very long time is going through ethical reviews, typically multiple rounds of ethical review in each country. Sometimes there are different organizations. And then even before we get our permits, we're also spending a lot of time speaking to the communities, explaining in layman's terms what we're doing, what the benefits are, if any, what the risks are, if any, and getting community consent. We translate into the local language. And it's only after getting those permits and community consent that we get individual consent, and then we proceed. We think it's really important to return results to participating communities. I think it's a form of benefit sharing, and it also lets them know what you did with those samples that they generously provided, which I think is really important. Training and capacity building is also really important, and I've had the honor of training a number of grad students and postdocs in my lab, most recently Tafadzwa Taffy, who got her PhD recently at University of Cape Town. So the kinds of phenotypes that we measure are things that you can do in really remote areas. So very detailed anthropometric phenotypes, height, weight, waist circumference, things like that. Cardiovascular, lung, and blood phenotypes like blood pressure and lung capacity and hemoglobin. We look at metabolic function, lactose tolerance, glucose tolerance, for example, and infectious disease status when we can. And then we're using an integrative omics approach where we're now looking at whole genome sequencing. We're integrating that with proteomic data, with metabolomic targeted and untargeted metabolomic data. We have gut microbiome data. We have gene expression from whole blood. We have gene expression at the single cell from white blood cells. Starting to collect epigenetics, not as much so far, but I'd really like to do that. And then we're looking at how those are influencing each other and also are being influenced by environmental factors, together influencing these variable traits and disease risk. So now I'm going to tell you about a study that we published last year in Cell, in which we did a high coverage whole genome sequencing study of 180 Africans from these populations shown here, who represent, if you look at the bottom here, this is an admixture plot where we're inferring genetic ancestry. Sorry, this pointer is not working great. I might actually just use this. Let me see. You know what? That's better. Much easier. So we're inferring genetic ancestry that's represented by the different colors. Each line is a person, and they can have ancestry from different ancestral populations. And so these are the African populations. You could see from the different colors, there's a lot of genetic variation, not just within, but between populations. And the red arrows represent the populations that we've selected for the study. So representing a lot of that diverse ancestry. Now, they also have very different subsistence practices, hunter-gatherers, pastoralists, agriculturalists, or some combination, very different cultures and so on. So in just a very small sample set, 180 individuals, we found 33 million single nucleotide polymorphisms. About 17% of those were novel at the time. And many of those are predicted to be functional. The majority of these predicted functional are in non-coding regulatory regions, but we have some in protein coding regions as well. From this data, we can make a phylogenetic tree. So at the end of each branch is a genome, and it's been color-coded by the language family spoken, and then at the end, we see what the populations are labeled here. And what we and everybody so far has shown is that the most basal lineages, the ones that sort of the most basal position here, belong to the Click-speaking San populations in Southern Africa, followed by the Central African rainforest hunter-gatherers. Then we have two Bantu-speaking groups that originated in Central Africa. We have the two Click-speaking Hadza and Sandawe from Tanzania. And these are predominantly Ethiopian populations and the Fulani, a nomadic pastoralist group from that we sampled at Cameroon. And then these are non-African populations, and you can see that they represent a subset of the diversity in Africa, and this is consistent with this African origin and migration out of Africa. So one of the things that we did is we looked at the ClinVar database, and we just wanted to see, are there variants that are predicted to be deleterious and or pathogenic, and they're not very common outside of Africa, or these are African Americans here. Maybe they're at low frequency in African Americans, but we find them to be common in at least one African population. We found 44 of these that were at high frequency in some of the African populations. So for example, this variant is a splice donor variant that's associated with a type of hypotension, very rare outside of Africa, but at about, looks like about 13% frequency in the Fulani. These are actually three different non-synonymous variants of this gene PD, I think it's I3, that's associated with a type of alopecia that's very common in particularly women of African ancestry. We could see in African Americans at a few percent, but in some of the African populations, it's quite common. So does that mean that it's not pathogenic, or does it simply mean that there could be modifier mutations? We don't know. This is something to look into. So then what we can do is we can compare these genomes to the reference genome and count up the number of places where they vary when you compare them. That gives you an idea of diversity. And we see at the very high end are the two San populations, followed by the rainforest hunter-gatherers. At the very lower end are the Amharic population from Ethiopia, who are about 30 to 40% admixed with non-Africans. And if we were to look at, say, Europeans, the rest of the world, they're down here. So there is a considerable amount of diversity in Africa. We then used a couple of different approaches, including at the top, we're showing the result of PSMC, which is only informative starting at, say, about 50,000 years ago to make inferences about the effective population sizes going backwards in time. It's interesting that we start seeing differentiation right around 250,000 years ago, but from 50,000 years ago going to the past, the largest populations with the largest effective population size are the San and the rainforest hunter-gatherer groups. And if we look at using SMC++, this is more informative for more recent time periods, we can see that there are other groups now that have much higher NE, so like the Amhara, the Mercy, Sandawe, Takari. And then we see evidence of a major population crash in these two hunter-gatherer groups, the Hadza and the Shabu. Both of them have a census size that's about 1,000. So then, together with our collaborators, Jeff Spence and Yun Song, we use a number of different approaches. For those of you familiar, I just mentioned them. We use F statistics. We use coalescent hidden Markov models. And then we use an approach that was developed by Yun Song's lab called MAMI, which is based on the joint allele frequency spectrum. And what is very important about that one is that it is considering models where you're not just looking at common ancestry, but you're considering migration as well and changes in effective population size. And that turns out to be particularly informative. So this is a schematogram that is way, way, way overly simplified. And when we took all of this and put it together, this was sort of the, let's say, best fit. But it may turn out not to be the only fit. It's a very, very complex history. So I'll just point out a few things that tended to be the most consistent. So one is that the model that when we use what's called a ghost population. What do I mean by a ghost? Not what you think of like a ghost. It's a population that doesn't exist today. These are lineages that we're detecting that are very divergent, would have been about diverged from the lineage to modern humans about 1.3 million years ago. That's kind of similar to what you might see between Neanderthals and modern humans. Very strong evidence for introgression, so admixing prior to the split of all modern humans. And then we see some evidence for introgression more recently in some of the populations like the rainforest hunter-gatherers and the Niger-Congo speaking populations. Second point, as I told you, prior studies have always shown, these are San, click-speaking San populations, have always shown those as an outgroup to other modern humans. But in this model, when we consider migration, for example, the best fit actually was a model in which the rainforest hunter-gatherers from Cameroon and the Southern African San share an ancient common ancestry about 80,000 years ago. And that it was implying that there was a population ancestral to those that gave rise to all other populations. The other thing you can see here is very deep split times in Africa. This over here, OOA, is the non-African population. So very deep splits, a lot of substructure for long periods of time. And then we see evidence of migration events, both recent and more ancient. So now for pretty much the rest of my talk, I'm going to be talking about our studies of how people adapt to different environments. And this is from a review paper. I think everybody here is familiar with the example of sickle cell, right? Mutation in beta globin. People who are heterozygous are protected from malaria. And so it stays common. Even the homozygotes in an African environment are going to die at a young age typically. Other examples might be lactose tolerance, for example, that evolved in East Africa and also in Europe. So with the whole genome sequence data, what we can do is use FST statistics, so a way to look at allele frequency differences. And we put these together. We do all pairwise combinations between populations, use something called the DI statistic. And basically what we're looking at, if we look at this more simple example, let's say we're interested in the BACA, and we want to know what's different, what alleles are very different frequency in the BACA relative to these two other populations. But these two other populations are similar to each other. Okay, so you're trying to find things that are very different in the focal population that you're looking at. Now we take this distribution, we take the top, I think it was like 0.01%. You have like, I don't know, 10,000 or something. And then you say, what are the genes nearby and what are they doing? Try to get an idea maybe of local adaptation. And when we do that, we find a number of examples. I'm just going to give you a few examples today for 3 hunter-gatherer groups. So starting with the rainforest hunter-gatherers from Cameroon, the genes that are very different frequency tend to be, or the variants that are different frequency tend to be near genes that play a role in, for example, bone development, chondrocyte differentiation. That's very interesting because one of the features of these populations is that they're very short-statured. It's thought to be an adaptation to a tropical environment. If we look at a different hunter-gatherer group, the Hadza, who live in Tanzania and have a very different environment, it's much more arid there, for example. It's really striking is we see this enrichment for variants that are very different near genes that play a role in heart muscle development. You could imagine how that could be adaptive in populations that are hunter-gatherers walking very far distances in very arid conditions. So we're actually following up on this now. Almost all of these are regulatory in nature. So we've identified a number of novel enhancers that are playing a role in regulating genes that play a very important role in heart development. So I thought today I'd tell you about a study. Frankly, I haven't really given this presentation much before, so I'm a little rusty on this, but I thought I might get some good feedback actually from you guys. So we were looking at association between genetic and environmental drivers of telomere length variation in ethnically diverse Africans, and we published this in American Journal of Human Genetics earlier this year. Just to remind you, I think everybody here knows that the telomere consists of tracks of tandemly repeated short DNA repeats, and it's associated with protective proteins. Telomere protects the genomic DNA through a number of mechanisms, but one is trying to prevent the ends of the DNA chromosomes from being degraded, for example, which would lead to a lot of instability. And it's been shown that telomere length plays a role in both health and disease. So for example, we know that the enzyme telomerase plays a role in elongating the telomeres. But if you have very, very long, excessively long telomeres, that can play a role in cancer, for example. On the other hand, if you have insufficient telomerase, and then you could end up with other problems, including I'm just reading off here, genomic instability, mitochondrial malfunction, and I think it can play a role also in immune response and things like that. So it has been shown that telomere length is a highly complex trait. Previous heritability estimates range from 44 to 80%. There have been hundreds of telomere length associated SNPs identified in GWAS, mostly from European ancestry individuals. It is thought that environmental factors, things like smoking and stress and other environmental factors, are influencing telomere length. There was this intriguing study that came out in 2018, and what caught our eye is that they were looking at people who've been infected with malaria caused by Plasmodium falciparum infection, and found that they had a shortening of their telomeres. And eventually, once they recovered from malaria, the telomeres got longer again. But this was a very small study. And we wanted to know, what is the relative importance of genetic versus environmental, including factors, including malaria, in influencing telomere length in Africans? And following up on an earlier study we had published where we had looked at a smaller number of individuals, we wanted to know, why is telomere length longer in Africans? It is significantly longer in Africans compared to other people of other ancestors. So our collaborator, Avi Avraham at Rutgers, using the old-fashioned, and he insists on doing this, Southern blotting technique, because as he says, it is challenging, but it is by far the most accurate. So a lot of people now use like qPCR to do this, but the gold standard, as he's always pointing out to me, is actually doing old-fashioned Southern blotting. And it's laborious, but we get really, really accurate results. So he did this in 1,818 individuals from the population shown here, so different regions in Africa. And then we integrated this with data from Illumina 5M array and from the H3 Africa array. And then we imputed missing genotypes from this PCA. The point is just to see there's a lot of genetic diversity here. So as everybody has noted prior, we see a correlation with decreasing telomere length as people get, as people get older. And as people have also seen significantly shorter telomeres in men relative to women. Now, what was also interesting is that we see significant differences in telomere length between African populations. I'm just going to point out a few things. The population with the longest telomeres are the San, those click-speaking groups that I told you have very basal lineages. The ones that have, let's see, I think I have it highlighted, the Fulani, who are nomadic pastoralists, who have some unique ancestry. They have sort of North African Berber ancestry and possibly some non-African ancestry, have the shortest telomeres. We can look at groups like, these are two groups. This is a group called the Mubukushu, who originate from Angola. We actually sampled them in Botswana. They're not admixed with neighboring groups. They speak a Bantu language. They originated from Cameroon, right? So we can compare them to the Takari Bantu speakers in Cameroon. And one thing you'll note is that these populations, these four, are from Cameroon in a highly malaria endemic region, and they have significantly shorter telomeres. And if we just compare these two groups with the same genetic ancestry, one's living in South Africa, Southern Africa, the other's living in Cameroon, the ones in Cameroon have significantly shorter telomere length. So that was intriguing to us. And this is, by the way, whenever I show you telomere length, we're controlling for age and sex whenever I show that to you. So we do a GWAS tiny sample size, right, around 1,800. So we're not going to see anything significant. But we can do estimates of heritability. And those estimates, I know it's hard to see, it's actually 0.66 is the SNP heritability. So very similar to what other people have seen. So, but I will show you just a few. They're not genome-wide significant. Is this? No, but it's close. It's like p10 to the minus 6. So one of these was at chromosome 13 within, can somebody tell me how to say this? Is it Cloto or Clotho? Cloto? Does anybody know? I think it's probably Cloto. I'm not sure. But what intrigued us is that there have been studies in mouse models where when they overexpress, okay, so in green is wild type, and here in these blue and purple are mice that overexpress this gene or this protein. When it's overexpressed, they have significantly longer lifespans. And by the way, I should have said also that longer telomeres have been associated with longer lifespans. And the variant that we found actually has been shown to be an EQTL. All right, so that's really intriguing. We find this variant associated at this gene that's been shown to play a role in lifespan. Another really interesting observation, we find this loss of function variant. It's knocking out the protein basically that is specific to the SON. It's only found in the SON. It's in this gene called PIF1. And that gene plays a role in regulating telomere length by preferentially removing telomerase from the ends of telomeres. And so what we have here is that the SON who have the loss of function variant, which you would expect, remember it, let me say, it inhibits telomere elongation. So if you knock it out, you would expect to have longer telomeres. And in fact, that's what we see. Those who have the peel-off have longer telomeres. And this is specific to the SON, so intriguing. Okay, so even though we don't have a lot of power for a GWAS, we can still apply polygenic scores, which we construct from the UK Biobank, where they did a GWAS of 472,000 participants using qPCR. And they discovered 197 associations at 138 loci, explaining about 4.5% of the variance in telomere length. So still not great, 4.5%. Of those 197 independent associations, 73 are present in our imputed data set. So now on the left, I'm showing you the predicted telomere length based on the polygenic score. And I will say that using these, the polygenic score from the UK, derived from the UK Biobank, it actually does not bad. I mean, it predicts about 3.2%. And remember, it was like 4.5% in the European, so not terrible actually. But what was like particularly remarkable to me is if you look at the predicted and the observed, it's very similar. And so the SON, for example, are predicted from the polygenic score to have the highest telomere length, and the observed telomere length is highest. The place where you start to see it deviating, again, some of these populations in Cameroon that have high exposure to malaria. So then we wanted to address the question, how do environmental factors correlate with telomere length? And so we actually, Mike McQuillan in my lab actually looked at like 22 bioclimatic variables, okay? And there are only two that come out significant, and they're highly correlated with each other, which is malaria and precipitation. Okay, so we think malaria is the driving force here. And we could see that as malaria incidence increases, you have a decrease in telomere length, even after controlling for things like genetic ancestry, for example. So we think that malaria is driving this. So Avi Abraham, he has a theory. I don't know if this is true or not, but this is his theory. So when you get infected by Plasmodium falciparum, it can result in breakage of the erythrocytes, right? So you have a lot of anemia, for example. So activity of telomerase is insufficient to prevent telomere shortening resulting from hematopoietic cell replication, which is one of the reasons we see shortening with age. His hypothesis is that children raised in regions of high malaria endemicity experience numerous bouts of erythrocyte destruction, which requires a very active amount of this generating new cells through this hematopoietic process. And he thinks that that is resulting in shortening of the telomeres. And depending on the intensity and frequency of malaria, these children may be more likely to become adults with short hematopoietic cell telomeres as expressed in LTL. But obviously, if you really want to get at this, you need to look at children, right? So we need to follow people over time, or at least look at different cohorts. So now in my remaining time, I'm going to talk about another adaptive trait, which is skin color. And in the upper left here, these blue colors represent melanin levels in populations around the world. We can see that it correlates with UV levels. So it's thought that when our ancestors migrated out of Africa and went to higher latitudes, there would have been selection for light skin color. It's been hypothesized perhaps this is to promote synthesis of vitamin D, which is D3, which is synthesized in the skin in response to UV. In areas closer to the equator where there's a lot of sunlight, you want to have darker skin for protection from damage caused by UV radiation, and perhaps also preventing folate degradation. So to infer pigmentation levels in the field, we use a spectrophotometer that we shine. In this case, we're looking under the arm because it's less likely to be exposed to sunlight. And we can, from the reflection of the wavelengths in the red spectrum, infer the melanin levels. We see quite a bit of variation in Africa. The most lightly pigmented are the San from Southern Africa, who I told you have some of the most basal lineages. And we see a lot of variation. And the most darkly pigmented in the world, actually, are people who speak a Nilotic language originating from South Sudan. But lots of diversity. So we did this GWAS, tiny sample size, relatively, only like 1,570 people. So we don't have a lot of power, but we did find 8 independent associations at 4 regions of the genome, and just going to quickly step through this. So the strongest association is a non-synonymous variant at SLC24A5. The variant associated with light skin, I'm going to always be showing in blue, and the variant associated with dark skin, I'm going to be showing in this sort of fuchsia color. Now, this actually was the very first gene identified in humans to play a role in skin color in Europeans. They found it, I think it was in zebrafish, and then looked in humans. So this was very well known. But what we wanted to know, we could see that it was common in East Africa. So did it arise independently in East Africa, or was it brought over by migration from non-Africans? So we do a haplotype analysis. Each circle represents a haplotype. How alleles are arranged in some, I don't know, like 40 kb region of the genome. The bigger the circle, the more haplotypes. And then, you know, these haplotypes are very divergent, let's say, from this one, and we color code it based on where the number of people from each region who have it. The take-home point here, there's this common long haplotype that you see in Europeans. It's been hypothesized that it's a target of natural selection. And in East Africa, they had that non-synonymous variant on the identical haplotype background. So that was probably introduced by migration. The next strongest association was that a gene that had no name at the time. It was just like open reading frame, whatever. It got assigned a name, MFSC12, based on homology to these ion transport proteins. And again, this is work with Bill Pavin and Stacy Loftus that we worked on together. And we were able to show that there are these two independent associations, one in an upstream regulatory region that's regulating expression of MFSC12, and then some within MFSC12 that frankly, we're still not sure exactly how they're acting. If we look at one of these variants upstream, we could see that the light-associated allele is very common in Europeans, East Asians, Southern Asia. It's common in East Africa, and it's very common in the lightly pigmented San, whereas the dark-associated allele is common in most other sub-Saharan Africans, South Asia, and Australo-Melanesia. So then Bill Pavin's group knocked out an agouti mouse at this locus, and it had a pretty dramatic impact on pigmentation. So we were able to show that, yes, definitely impacting pigmentation. Even Jewett and Yun Song, our collaborators, constructed gene genealogies. Think of this, if you were to unwind this, think of it like that phylogenetic tree I showed you, but this is looking at gene genealogy. So we're looking at each end is the sequence at this gene. It's very hard to see, but there's an open circle here. That means it has the light associated allele. And if it doesn't have that open circle, it's the dark associated allele. And we're looking at when there's coalescence of these lineages to a common ancestor. So a couple of interesting things. One is that the ancestral allele is the light associated allele. And secondly, that both alleles have a very deep time to most recent common ancestry. So they've been segregating for about 1 million years. The next third strongest association was in this region near this gene DDB1. We actually thought that was a candidate for a number of reasons, one of which is the fact that it plays a role in DNA repair after exposure to UV. People have mutations in this gene or in genes coding for proteins that interact with DDB1, have a disease, xeroderma pigmentosum, can't be exposed to sunlight because you can't repair the DNA damage. You're prone to getting skin cancer. And then I looked it up and I saw that it also is causing the color of tomatoes. So I'm like, it's playing a role in pigmentation. I bet this candidate. And there's some very early chromatin interaction data that suggested that our two independent regulatory regions we found here were interacting with DDB1. I'm going to come back to that in a minute. So we used this test of neutrality based on, it's called Tajima's D test, which is based on the allele frequency distribution. So what I mean by that, if look in a population at the number of singletons, doubletons, tripletons, and so on, this blue line shows what you expect under neutrality. If you see an excess of rare variants, that may be indicative of recent positive directional selection. If you see an excess of common variants, that can be indicative of balancing selection, maintaining variation. And this was done by a former postdoc, Xiaowang Dan, and we were looking at the 1,000 genomes populations here, and then also the San and Anilus Saharan group. And what we see is that in the Chinese and the or the CEU European population, there's evidence of a very massive selective sweep there. Over hundreds of thousands of base pairs that overlaps. Here's DDB1, I think it's right around here. And in fact, if you look at the heterozygosity in that region, it is wiped out in Asia for hundreds of thousands of base pairs. That's what you expect if a variant really rapidly rises to high frequency, it wipes out all the neighboring variation. We see that not quite to the same extent, but in Europe, and you don't see that in Africa. And when you look at the haplotype here, this is actually over 200,000 base pairs. There's this really long haplotype outside of Africa. And we think, and then if you look at the gene genealogy, there's this rapid coalescence of all the non-African lineages, or many of them, right around 60,000 years ago. I think it's a really rare example that when these chromosomes were introduced out of Africa, they swept almost complete, a major sweep outside of Africa. Was it for pigmentation? Not necessarily. There's a lot of LD, and there's many important genes in this region, so we don't know for sure. The last wouldn't be a surprise to anybody, right? OCA2 and HERC2 are known to be associated with pigmentation. So mutations in OCA2, for example, play a role in some of the most common forms of albinism. And we found these two variants in this upstream HERC2 region that were in non-coding regions, and we showed that they influence expression of OCA2. We saw a non-synonymous variant in exon 10 of OCA2. It was not correlated with gene expression, but it turns out that's correlated with alternative splicing, and that people who have the shorter transcript, which happens to be missing all of exon 10, and but it's still in frame, it still codes for a protein, but that protein is missing the third transmembrane domain. So you can imagine it's having a major impact. And what was, I thought, really cool was that the only place, for example, where we see darkly pigmented alleles outside of sub-Saharan Africa is South Asia, Australo-Melanesia, places where we see darkly pigmented people. And we actually were able to show that those are on the same haplotype background as in Africa. They were introduced by the migration out of Africa. So in half the cases, the ancestral allele was the light associated allele. And most of the derived alleles originated before the origin of modern humans. So what this means is that we don't know what the ancestral state was in Africa, but chimps actually tend to be lightly pigmented because if you have a lot of body hair, it may not matter so much, right? But as our ancestors left the forest, went to the savanna, there's going to be selection for thermoregulation to lose body hair. You're going to have your skin exposed, there'd be selection for darker hair. But I wonder if there was a lot of variation even in the past, and that both light and dark skin has been evolving in Africa. Okay, now going back to what I told you about before, these signatures of selection, now we're going to look at the third hunter-gatherer group, the San in Southern Africa. And what we see here is that genes that are very different, that have alleles that are very different in frequency, are near genes that play a role in pigmentation and hair follicle morphology, which is interesting to me because they have a very unique hair type. And even things like narrow eye opening, it's another unique phenotype in this population. So now we're following up on these. And this was published earlier this year in Nature Genetics, led by a really talented postdoc in the lab, Quan Chen Feng. And what you can see is, I get this every time I submit one of my grants, you don't have a large enough sample size. You don't have a large enough sample size. We will never, ever, ever, ever have a sample size that competes with the European populations. I say, so what? Do we just ignore these populations? No. And there's information there. So if I just say, I'm going to ignore the standard genome-wide significance level, and I'm just going to take the top 5,000 associations and then ask the question, what are the genes nearby and what do they do? Well, they are strongly, strongly enriched for pigmentation. So there's biologically important information, even if it's not at genome-wide levels of significance. So we want to go dig a little bit deeper. So we take these top 5,000 associations from GWAS, and we take all these DI selection scan hits from the lightly pigmented San. Then we narrow these down to 1157 candidate SNPs that overlap predicted open chromatin regions that would be active in melanocytes, which is the cell type that you would care about in this case. And then we use a massively parallel reporter assay where we can make oligos that have, let's say, the dark associated or the light associated allele. You put them into this lentiviral vector that has a barcode. You then transfect these melanoma cell lines, and then you do kind of like an RNA-seq analysis, right? And so you could see how they are driving gene expression. Now, we do this in two different cell lines, both darkly pigmented and lightly pigmented. And as it turns out, it's a good thing we did that because you don't always get the same result, probably because of the different trans background in these different cells coming from people of different ancestries. About 45 overlap. These are looking at regions where there's a very significant difference between the expression of, say, the light and the dark associated allele. But some of them are specific to different cell types. So we both want to identify potential regulatory regions, but then we want to know what are the target genes. So we generated Hi-C maps and also H3K27 acetylation chromatin maps to help us identify what the target genes are. Then what we did is we either use CRISPR inhibition, in which the CRISPR-Cas9 inhibitor binds to the enhancer and represses the activity of the enhancer and the promoter, or you just chop out the enhancer. Okay, so that's the enhancer knockout. And I'm just going to give you a couple of examples. This one I was really excited about because this was actually identified from the selection scan. And it's at almost 100, it's at pretty much 100% frequency in the sun and also in Europe and basically all non-Africans except for Australo-Melanesians. And this, I had seen this gene before. Actually, it came out in a study looking at genes under selection in rainforest hunter-gatherers and associated with height. But what we found here was in a different region, okay? And it's in a regulatory region. When I go to 1,000 genomes and we calculate FST, it's extraordinarily high, okay? This is highly, highly differentiated between populations. And what we can see is this is the candidate variant that was identified by scans of natural selection. It's in a predicted enhancer. We could show through chromatin interaction that it's interacting with the promoter of TRPS1. Here we did a luciferase expression assay just to replicate, and we show that the C allele that's associated, we think, with lighter skin has decreased levels. When we do CRISPR inhibition, we can see decreased levels of expression of that gene. And in addition, what's cool about this is you can actually measure pigmentation in the cell line. If you look at MNT1, the darkly pigmented cell line, and we see that when we do CRISPR inhibition and there's a decrease in expression of TRPS1, we see a decrease in melanin levels as well. So it's showing that it's impacting the phenotype. Now I want to come back to DDB1. And the strongest GWAS association in that region, and our strongest candidate here, was about, I think it was about 40 kb or so from DDB1. Very close, though, to these genes, TMEM138 and CYB56183. And what I want to show you is that in this case, the T allele is associated with darker pigmentation, okay? And the A allele is associated with lighter pigmentation. And what we see here, when we look at this reporter assay, is that the T allele shows lower expression. And remember, I told you the T allele is the dark, associated with darker, so it's the opposite of what we've seen before. When we look at where the chromatin interactions are, they are most strongly with these neighboring genes, CYB56183 and TMEM138. There is some interaction with DDB1, but it's not quite as strong as what we're seeing for the other genes. So now we do the CRISPR inhibition shown here, and we do the CRISPR knockout shown here. And we see a significant decrease with inhibition for the TMEM138, CYB56183, not for DDB1. When we do the enhancer knockout, we do see a significant decrease for all, but you can see it's not quite as strong for DDB1. We can then, after we do the knockout or the CRISPR inhibition, you can do RNA-seq, look at changes in gene expression. The ones that are most strongly decreased in expression are the TMEM138, CYB56183. So we think that these are probably the more important target genes. It doesn't mean DDB1's not playing a role, but we think these are really critical. Now, when we knock it out or use CRISPR-I, we do not see a decrease in melanin, and that's because, remember, I told you when they're knocked out, you see darker, but we're using darkly pigmented cells. So what you have to do, overexpress the protein. If you overexpress the protein, now we see a decrease in melanin. Okay, so we're showing that it is impacting, both of these seem to be impacting pigmentation. So now, if we look at overexpression, and I think down here we have the knockout, and we look at gene expression, we can see that the genes that are altered in expression levels, many of them are very famously known to play a role in pigmentation. It also impacts things like melanosome development. I think there's mitochondria and lysosome here as well. I'm having a hard time seeing because it's so small. Now, if you look at where these genes are expressed, CYB56183 is very highly expressed in the skin. TMEM138, not so much so. So we were very interested in CYB56183. Our collaborator, Elena Awancia, looked at where this protein is located, and she shows that it overlaps with the location of TYRP1 in melanosomes, which is where you would expect it to be if it's playing a role in pigmentation. So what is it doing? I have no idea. So I just looked this up and I see that it's a transmembrane reductase. It reduces iron cations. It is active in lysosomes. And as we know, lysosomes and melanosomes have some similarities. So we suspect that it may be involved in endosomal and lysosomal and maybe melanosomal cellular iron homeostasis. So then I start looking up papers. What does that have to do with pigmentation? I find this, it says iron upregulates melanogenesis in these cultured retinal pigment epithelial cells. And that when people have infusions, intravenous iron infusions, that they have this hyperpigmentation phenotype. So I think there could be something going on here. But my collaborator, Mickey Marks, is actually really interested in this other gene, TMEM138, which plays a role in development of cilia. And I found this paper saying that cilia can play a role in melanogenesis. And he's actually following up doing some cell biology studies of this gene. So in conclusion, there's an urgent need to include more diverse people in human genomics research. African demographic history is very complex. High levels of genetic and phenotypic variation in Africa are very informative for genome-wide association studies. Integration of GWAS with signatures of selection are informative for identifying functional loci. And functional genomics is very important to actually identify what the causal loci are. With that, I will just end by thanking the many people involved in these studies and the funding organizations, and glad to answer any questions that you may have. Yeah, you can do this one.
First question was, just wondering, are these interesting African genomes being made publicly available in the public domain?
Yes, they are. So yeah, they're all available through dbGaP. Yep.
Thank you for the talk. I have a question about the telomere length study. So what exactly was done to exclude the effect of aging when doing telomere length comparison? And can it be that the speed of telomere length decrease? So aging is different between different populations, not only the length of the telomere itself, but the speed of the decrease.
So again, we can control for that. We can take that into account as a covariable, right? So when we do that, that alone certainly can explain what we're seeing. So we can show that there is a correlation between age and telomere length, but that alone as a variable is not explaining what we're observing. And when we put it in the model with malaria, for example, and other covariables, it is only malaria that comes out as being causal. But those are some interesting questions. I think it'd be really interesting to do further studies and look at things like differences. Frankly, it can be very hard to look at these questions about lifespan. In groups, I can tell you, just anecdotally and from what I've read, the San, for example, do have very long lifespans, actually. They have the longest telomere length. But in the rainforest hunter-gatherers, they have such a harsh lifestyle that the chance of living to age 15, I think it's like 40%. If they make it to that, the expected lifespan is like 24 to 25 years of age. They have a huge amount of infectious disease and things like that. So that's a problem. It's very hard to study in those populations where they are dying for many other reasons in that harsh environment.
Thank you. And unrelated second question, I've noticed that Madagascar was excluded from all that map. So is it not considered when they're doing African population studies, or it's a completely different story, or it was just the case on that?
So some people are studying African genetics. I'm forgetting, who is that? PJ Perry, maybe some others. Oh, Himala Sijal, I think, was looking at them. I know people are looking. We do know that that population is very admixed. So people of this Bantu-like ancestry, but we also have people from like Southern Asia coming in, Southeast Asia. It's a very, very admixed population. Didn't go there. I would love to go there, frankly, but can only do so much. So we haven't been there yet. Yeah.
The next question from online, while you're getting the mic. I wonder if those studies investigating telomere length with malaria factored the influences of malaria treatment drugs.
Wait, say that one more time. Oh, wait, where is it?
Factor in malaria treatment.
Oh, no, but I can tell you that the populations that we're studying are very, very rural. They do not typically have much access to healthcare. They are endemic for malaria. So we did both thick and thin blood smears. We also did a rapid test for malaria. We actually, from genome data, know who has Plasmodium. And so it's just kind of endemic in the population. Most of them are not getting treated for it. Unless they were acutely ill. If they are very acutely ill, then they might be. But the adults often have some immunity to it. Yeah.
Thank you so much for this research. Question just following up on doing sequencing, and I've mostly worked with African-American populations, but are you using short read whole genome sequencing, and what reference panels are you using, and do you have recommendations on things like that?
So for these initial studies, yes, short read sequencing using Illumina, and just doing what everybody else has been doing, mapping it back to the reference genome. So no doubt we are missing really important regions, structural variants, or things that might be we have plenty of unmapped reads that are not mapping any, right? So we know, and others have shown this. So we're starting to do some work looking at using long reads to generate better reference genomes in these populations, and others, people here, right, have been doing this. And so I think as we start there are efforts by NIH to get more reference genomes. And so then we can make this pan genome, and then we can use that for mapping the short reads. So I think that's going to be super informative.
Okay. And do you think with the sequences that you have currently, can that be helpful in looking at local ancestry and things like that to learn more about the data we currently have with African Americans?
Yes.
To identify like areas of Africa they might.
Yeah.
Thank you.
Hi, I really enjoyed your presentation. I had a question about the telomere studies.
Yeah.
Do you get information on the parents of the individuals that you are sampling? I wondered if the age of the parents was at all like taken into account for things, because I know in some studies they see that the father's age can have a strong impact on the telomere length of the offspring.
Oh, that's interesting.
So I was wondering if that had been taken into account.
No, because it is very challenging, for example, to do traditional pedigree studies in these populations. Frankly, everybody's welcome to participate. And so often we do have related people. So we do have knowledge of who's related. And for things like when we're estimating heritability, essentially you're taking that into account, right? You're looking at like a relatedness matrix. But I can't say for certain what that age is. And also, we do questionnaires where we ask questions like age of, when you gave birth and how many children, this and that, but it would be challenging, I think, in that environment. I think it's a really interesting question to follow up on. Yeah, thank you. That's really interesting.
Fascinating talk. I'm curious about the ghost populations.
Mm-hmm.
Can you say anything about those populations from the sequences that you have? Or, of course, do we have any clue about what those populations were?
Unfortunately, no. We can't see anything, because keep in mind, without a reference genome, right? So for Neanderthals and Denisovans, there are reference genomes. That makes it way, way easier to go into the modern genome and say, yes, that region is from a Neanderthal, or yes, that's from a Denisovan. We can't so easily do that here. With the approach that we're doing, we weren't targeting particular regions. We are doing things right now, like doing sliding window TMRCA across the genomes. We're going to identify regions that are highly divergent. Sometimes there can be other reasons. There could be balancing selection, things like that. It can be very, very tricky to pick out which ones are due to this integration without having a reference. And for that reason, we don't know. I'm just waiting for someone like David Reich or Svante Pääbo or somebody to actually get the genome sequence from a really old African, like 50,000 years or older. I'd be so thrilled. Many people would, because it would tell us so much. I mean, once we have that reference genome, there's some really cool things that we can do. Although one thing that we have been doing is comparing regions of the African genomes that are homologous to Neanderthals. And by doing that, we actually had a study where we were able to show that there is sharing in every African population regions with Neanderthals. And the reason is that we believe there was a very old anatomically modern human population, could have been from around 250,000 years ago, that left Africa and interbred intergressed with Neanderthals. And you can actually see interesting patterns of like, these deserts in the X chromosome where you would have, there seems to be selection against having both the anatomically modern human and the Neanderthal sequence. So using those sequences, we might actually be able to infer something. If you find enough of those, you can actually reconstruct maybe a genome from this early ancestor. It's something we're trying to do, we're thinking about, but it's a little tricky. But that would not be the intergress population anyhow. That would be early modern human.
Okay, last burning question. While you were visiting these African countries, did you pick up any languages that you can say you
DIR.
fluent in?
Nina se Kiswahili kidogo. Just a little bit. I'll embarrass myself because so when I did a postdoc years ago with Andy Clark at Penn State, and at that point I knew my passion was to do field work. And I said to myself, I better learn how to speak Kiswahili because our focus was going to be in Tanzania. And so I took a year of Kiswahili. And when I was there, I can just say things like introducing myself and, mimi ni mwalimu, I'm a teacher. I'm really stretching to remember all my Kiswahili because if you're not there and practicing it pretty regularly, you start to lose it. So I want to go back there and practice. I want to apply my Kiswahili. I find when I go back, it starts like coming back to me a little bit more. And people do appreciate, even if I'm not that fluent, they appreciate that I'm trying. They appreciate that.
And a minor question, in one of your slides where you were talking about the melanin data, there was a line there that says diluted coat color. What do you mean by that? One of the Go terms.
Oh, it was the mouse phenotype database.
Okay.
Yeah, and frankly, which turns out to be much more informative for all of the pigmentation related phenotypes, because we don't know as much. We know more from the mouse than we do from human. Yeah. My pleasure. Thank you.